Okay. So I posted the second homework this morning, and it's due next Friday. So not this Friday, but next Friday. And this will be relevant to stuff that is in a new script that we'll start working with today. So on eCampus, there's a code file called seekanalysis.r. And this will be about doing manipulation and exploring and some analysis on sequence data, DNA sequence data. We've already talked a little bit about where sequence data comes from and what it represents. Now we're going to say we have sequence data. Let's explore large-scale characteristics of it. Let's also learn something about how to align multiple sequences to each other. That's a challenging task. For example, if we were working with humans, we have in a database online somewhere, we have this sequence of one or more human beings who have been entirely sequenced. We have the entire ACTG code. And so if we were to obtain sequencing information for a new person using, say, the next generation sequencing stuff that we've talked about, we would get these millions of short little sequences, A's, C's, T's, and G's, and we want to map those back to a reference genome. Basically, I want to find for each of my little reads, where did this come from in the human genome? And if everybody's DNA was exactly the same and there were no errors in the sequencing procedure, there were never any mutations or anything like that, it would be relatively easy to do that. You would have a sequence of A's, C's, T's, and G's, a short sequence obtained from an individual, and you would just find where in the reference genome does that happen. That's where I map it to. In the real world, though, there are differences in our sequences. Yes, question?
So you were talking about the errors. So is that related to genomic errors, like the errors that you related to us, or the errors that are related to the map?
Okay, so question was, when I said errors, am I referring to like mutation issues where my DNA looks a little different from yours because of mutation, or about errors in the actual, like the software and the laboratory equipment taking a tissue sample and turning it into A's, C's, T's, and G's? The answer is there can be both of those going on. There can be mutations that represent bona fide differences in the DNA sequence between two individuals, even though they're the same species. But there will also almost certainly be some of your A's, C's, T's, and G's in your data set that were incorrectly classified. Hopefully not much, because we will employ some kind of filtering on quality scores. For every sequence that we get from these technologies, we will get some kind of number that quantifies, was this a really confident A, or could it have been a T, you know? And usually you'll employ some kind of filter to say only keep the reads that have really high quality, believable sequences. But still, there is the possibility that here's my reference genome for human, and here's my pile of reads for some human, and we're going to try to find where they line up in the reference. There's the possibility that a read goes in a particular place, but one or more of the letters in the read were incorrect from the sequencing technology or from the algorithm that was used. So the process of mapping or aligning a short sequence to a big one has to basically find where's the best, if not perfect match, where's the best match. and hopefully give me some kind of number that quantifies how confident you are in that match or alignment. So I'm going to talk a little bit about what happens when you ask to do an alignment, because when you get your spreadsheet full of numbers and there's a number next to mouse 12 for gene X, that number is dependent on our having correctly put the observed data in that slot. If I have a bunch of reads and I'm going to say these reads came from gene X, somewhere behind the scenes there had to be an alignment of that pile of reads to gene X. So we want to be confident in doing that. I'll talk a little bit about the algorithms that are used for alignment. So that's what's coming up. We'll spend a few lectures on this material. The next material will be more data analysis, like here's a bioinformatics data set, an M by N matrix of numbers, and let's start doing making pictures and exploring and doing statistics on it. All right, so homework 2, there's a new package that we'll play with starting today. It's kind of like BioMart. I like it a little bit better. I haven't had the issues with this one that we've been having with BioMart, like bad connections or that kind of thing. But it allows you to do similar things, download sequence data. You'll do a toy example of aligning two sequences. So what's the optimal way to put these two short sequences next to each other? For example, we had a sequence GAG and a sequence GTAG. So things we could do. We could say we could line them up just as they're written. GTAG, in which case we're saying there's a match, there's a mismatch, and there's a no match. Another way to line them up would be like this. Let's put a blank and then line GTAG down below like that, in which case we have a match, 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 and just one issue where there's looks like maybe a deletion in the first one or something like that. So the alignment thing you're going to do is to use an algorithm. You will define a way to reward matches and penalize mismatches, penalize gaps like that one. Basically, if you make a decision on how to score things, it's just a computational problem. Find the optimal way to put those things together to maximize the score. So you'll get practice doing that here. You'll get practice doing it with a protein or an amino acid sequence. It's the same idea. It's just a little more complicated because there's more amino acids than there are nucleotides. Okay. And then the last question will be kind of getting us ready for the next topic, which is an M by N matrix of numbers. So you'll download an example data set. These are proteomics data. So the data will be numbers for each of thousands of peptides in this case. I'll talk about it more, but if I want to study proteins using, say, mass spectrometry, which is a commonly used technique for proteomics, I'm actually going to get numbers for peptides, which are pieces of the proteins. The intact proteins aren't amenable to this technology, so you'll get your features, the rows in your M by N matrix will be peptides. So download some example data and make some pictures. We're going to start exploring bioinformatics data. And these will be simple pictures like box plots, side-by-side box plots. And then you'll do our first actual statistical test. Actually, you did that with the Go enrichment, but we're going to do a t-test, feature by feature. In these data, there are comparison groups. So you'll learn about it when you read the problem, but you're going to compare two of them, high concentration, low concentration, do a t-test, and ask peptide by peptide, are you statistically significantly different in your abundance between these two concentration groups? Show me a histogram of the p-values, and that's basically it. Okay. Let's see, before I move on to our next set of code, I just wanted to share a colleague of mine on campus. She's in the neuroscience department, is putting a grant application in right now. And it's relevant to what we're talking about. We have talked some about this epigenetics idea, the idea that while your DNA sequence dictates what you are, there are mechanisms where your body will do different things depending on the circumstances, the situation, without any changes happening to your DNA. So same reference manual, same code, but depending on, are we starving, or are we sick, or have we been exposed to radiation or something? Depending on external circumstances, different genes will turn up, turn down, turn on, turn off, and so on. That's the idea of epigenetics, where there are kind of a supporting cast of molecules and mechanisms that can regulate what other genes are doing. Okay, so what this project is about is studying Alzheimer's disease. And Alzheimer's disease is an issue of your brain. So let's say you wanted to diagnose somebody. You're exhibiting signs of dementia, you're about that age, it's possible you have Alzheimer's disease. If you really wanted to diagnose the Alzheimer's disease, as far as I know, you would need brain tissue to look at the brain to see characteristic physical manifestations of it. So it would be really nice if you didn't have to do that, because that's pretty invasive. And what the project is about is taking a blood sample and looking for RNA, a particular type of RNA. As far as we know so far, RNA is there because it copies DNA, copies genes, and then the RNA copy goes off to the manufacturing plant where proteins are created. But I've mentioned a couple of times that RNA can do some other things as well. So RNA can be involved in regulating other genes. It can play a role in this epigenetics idea. In particular, there's something called microRNA, which is short or small, relatively small RNA. It does not code for protein. And so up till relatively recently, we wouldn't have known what it did, if it did anything. But it does do something. What it does often is regulate the expression of other genes and often turn them down. So if there's microRNA that's specific to a gene, what will likely happen is when that microRNA are expressed, when they're put out there, they will turn down the expression of their target gene or genes. They will regulate the expression. And there's a particular type of microRNA called circulating microRNA that can survive outside of their target region. So you have a cell, inside the nucleus of the cell is your DNA, and it never leaves the nucleus. RNA copies are free to leave the nucleus and go to the ribosome and turn into a protein, but most of the time everything stays inside the cell. There are these circulating microRNA that can leave the cell, and they might just be floating around in the blood. And so the project is about, we know apparently that there are certain microRNA that show up when Alzheimer's is happening, I guess. And the thought, preliminary data from mice. So you want to know about people. That's what they're most interested in. Human beings walk into the clinic exhibiting signs of dementia. We would love to be able to prick your finger, take some blood, and confidently classify you. I'm 85% confident you have Alzheimer's disease or something. Would be great. And these circulating microRNA are tough enough that they don't get degraded or fall apart in the blood. So if there's issues in the brain where a microRNA-mediated regulatory mechanism is happening, and if some of those microRNA make it into the bloodstream, then they're floating all over the place, and you don't have to go to the brain because representatives of what's going on in the brain are in the blood. So take a blood sample and filter out the microRNA and then do t-tests and stuff, or make a predictive model that we'll talk a little bit about. I want to predict or classify based on an individual's microRNA profile whether they have Alzheimer's or not. So I thought it was a relevant and kind of interesting project. Okay, so I'm picking back up with seqanalysis.r, and we are going to retrieve a sequence. We have done this using Biomart. We're going to use a different package this time. So I am going to install the seqnr package, BioClide seqnr. And then we will download a sequence. So again, the first bit of code here is a reminder of what we've already done. You can use the Biomart package to get a sequence. For example, if we wanted the DNA sequence for the BRCA1 gene, this code is like what we saw last week. I'll go ahead and run this as a reminder. Actually, I won't. You can run that. One more caution about Biomart. I can't remember the exact sequence of events that causes this to happen, but I have had it happen in previous semesters that, for example, if you library Biomart and you do something, and then maybe you library seqnr and try to do something else, they might compete with each other. For example, I think both seqnr and Biomart have a function called getSequence. I have had issues before with loading Biomart and then trying to move on and do something else. So I would recommend, if you're going to work on homework 2 that's going to involve seqnr but not Biomart, don't load Biomart unless you need it. And if you get some weird error after having loaded Biomart with some other stuff, I recommend closing R, starting over, and only using the other stuff. Biomart is finicky. Okay, so how's the installation going? I will go ahead and update all of them. Answer's yes, right? Okay, that seemed to have worked. Let's library seqnr in. Okay, so yeah, I had already loaded Biomart. This is not really a problem, but it is giving me this message that that object is masked from Biomart. All that means is there's multiple objects of the same name. And what R will do in that circumstance is use the most recently loaded one. whenever you library something in, it's like when you open R, you have all the functions that come with base R. So like plot and mean and hist and all those. So when you type hist, it looks and it notices that it has a function. When you load in Biomart, you get access to all the functions and data sets that came with Biomart. When we loaded in or libraried seq in R, same thing, we had access to everything. But now there's a get sequence from seq in R and a get sequence from Biomart. If I were to type get sequence, R is going to use from the most recently loaded collection of things. So FYI. Seq in R gives us access to databases with sequencing information in them. We are going to, here's Ensemble that we've met before, several others. We're going to use this one, GenBank. GenBank is an online repository for sequence data. So I will say choose bank, GenBank. Okay, and now I'm ready to actually use seq in R. So I want sequence for BRCA1 gene. The main workhorse for seq in R is the query function. So I am going to $query. If you see a file like this, when you do function name, say, this is telling me there are multiple functions named query in R. So we have this issue again. The one that I'm interested in right now is in package seq in R. So I would like to see the documentation for seq in R's query function. So I'll click on its link. And then we have a familiar looking help manual. The arguments. Okay, so I'm going to specify the names of the objects I want to look up. I'll show you how to do that in a second. This requires that we use syntax that is specific to seq in R. I will show you some examples in a moment. Okay, here are the pieces that we can use to build a syntax. So go look up, say, BRCA1, no, that's going to be a name that I provide for the resulting object. This is really where everything happens. So this is where I'm going to say, look up BRCA1 from this specific organism and anything else that I want to specify. I can do that through these pieces. Looks like, in a simple case, this right here. So query, where the actual query is, species is Homo sapiens, and symbol, if you look up the help file, K is keyword. So I have BRCA1 and I want to look that up. I think I'm following what was in the textbook as an example or in the help manual for seq in R. If you have an H, G, and C symbol, I think you can use the keyword argument. And I specified the species through the SP argument. So if I do this, sometimes takes a few seconds to run. All right. And then we can investigate what came out. So there's two functions that come with seq in R. BRCA1, this thing that we just created, is another specialized, or an object of a specialized class. So if I type BRCA1, I get some kind of cryptic description of what it is. And I'm going to need to use the specialized functions from seq-int-r to actually interact with it. Two of those functions are here. Give me the sequence and give me the name. So let's run these two commands and look at them. And what we're going to see is that we have many sequences. We asked for the sequence for BRCA1. That's a single gene, single location in the DNA. Start here, stop there, that's BRCA1. But we've talked about the fact that there can be different versions of the same gene. So we actually have sequences for 163 things that are called some variant of BRCA1. Length. First of all, what is the class of BRCA1-seq? It's a list. Remember what a list is? It's a flexible way to store multiple things. Each entry can be its own object of whatever class. And what's the class of BRCA1 names? Character. Okay. What is the length of BRCA1-seq? Should be 163. So I have one element of the list for each of the sequences that came out. Here's the first one. If you want to index one element of a list, like here is a list. List one, I'll do the number 1, the NA character, character string. We'll do that. So X is a list, and it has 3 elements. All 3 of them can be whatever they want to be. If I just want to pull out the third element, I can do that by using double square brackets. If it were a vector, I could just use single square brackets. But with a list, I pull double square brackets. If I just do 1, I still get the same thing, but it's still recognized as a list. So if I just want to pull out the third element, use double square brackets. Okay, so let's look at the first sequence that came out. BRCA1 sequences, give me the first one. Looks like that. And give me the first name. So what is the name that's associated with that? That. All right. So we have different sequences that came from the same region of the DNA that's collectively known as BRCA1. Each one of these sequences has been identified as doing its own thing, roughly speaking. So here is one version of BRCA1. If you were to code that into a protein, it would do something. But there's a whole bunch of other ones. Related to that topic, I wanted to show you this picture. I've mentioned this idea of one gene going to multiple proteins. A buzzword that you might hear if you don't already know it. I want to go back. Is alternative splicing. So let's look at the picture. Up here on the top is one region that is called a gene. So imagine this is where BRCA1 lives. If you go to this specific region of the DNA and you start right there, or start right there and start reading, that is BRCA1. We've talked, though, about the fact that there's some pieces of that sequence that are needed to actually code for the protein, and there's other pieces that aren't needed. There's these introns and exons. And when the transcription process happens, that's turning DNA, this region of the DNA, into a copy of RNA. When that happens, you get a copy of the entire piece. And in this case, there were 5 exons for this gene. We're playing pretend. Pretend there's 5 exons. So to actually translate this gene into a protein, the full version of it would require that I pick up that intron and, or I'm sorry, that exon and that one and that one and that one and that one and stick them together. And then read off the sequence and turn that into a protein. So that might look like this right here. I have the blue exon, the green one, the red one, the blue, and the yellow one. Translate that, you get a protein. But depending on maybe the external circumstances, your body has the ability to not necessarily use all 5 of these exons. So here is another possible protein product of the same gene. And what happened was exon 3, the red one, was not included this time. So it's very similar to what we had over here, but there's a piece missing, and that is going to result in potentially a totally different protein. And here's yet another one where we took 4 out of the 5 again, and we got a different protein. So that's referred to as alternative splicing. And what we have in this BRCA1 output is sequences for lots of versions of BRCA1, 163 versions. So if you scroll through here, there's all these different sequences. These are all subsets of the whole thing for BRCA1. It should be 163, not 136. The very first entry here, BRCA1 names, here's the first 10 of them. The very first one is given this name. And so let's learn about that. What do we know about this particular version of BRCA1? I looked it up. There's a website that we'll go to 2 or 3 times over the course of the semester. It's here, NCBI. So I'm going to go to ncbi.nlm.nih.gov. The URL is in the script. Takes me to the National Center for Biotechnology Information. And I'm going to search for the name of that first variant, AB6-21825. I'm just going to put it right there. Lots of things that I could look at. I'm going to click on gene. I'm looking for gene sequence information. So tell me about the gene that's coded with that name. So I'm clicking on gene. And I get information here. So it is BRCA1 big picture, but then there is some more specific detail to this version of BRCA1. And we'll come down somewhere in a moment, and I'll show you how to download a particular type of file, FASTA file, for this sequence. In a moment. Yeah, so FASTA. I'll tell you what that is in a minute, but we're going to download a sequence in a particular format. Okay, so if I scroll down, I'm looking for the actual sequence. I think the display has changed a little bit since my last time here. What I was going to show you is that the sequence that came out of R, BRCA1, seeks 1, is like 71 bases long. If you look up this code, you'll see more than 71 bases, which is a little confusing. Confused me the first time I did this. Somewhere on the NCBI site, there's the actual sequence for this gene, and it doesn't match exactly what just came out here. So one other comment that I wanted to make about sequence data is they do match. It's just that what seq-in-R is showing us is the portion that actually turns into protein. There's a longer sequence that is given this code, but only the last 71 bases are actually turned into protein. There is another buzzword. I'm trying to limit the buzzwords we encounter because we don't need to know all of them. But if you look in some of these files that contain sequence information, you might see something called CDS. And if you see that, if you see a C, think coding. CDS is referring to the coding region of a gene. So if the BRCA1 gene has 5,000 A, Cs, Ts, and Gs, but the variant that we're looking at only involves the last, say, 71 bases, then it's 5,000 bases long, that's the gene, but the CDS, or the coding region, is a subset of it, and it's only the last 70 so. All right, so we have used seq-in-R to download the sequence for BRCA1, and let's keep moving. So I'm going to close that connection to GenBank. The reference manual says it's always a good idea to close the connections when you're done, because these can be eat up your memory, I guess. Wanted to show you what a FASTA file is. These are text files that are standardized ways to bundle sequence data. And depending on what kind of application you're working on with bioinformatic data, you might be given FASTA files to work with. If you do next generation sequencing data, for example, there will be FASTA files behind the scenes somewhere. So it's good to know what they are, and that Bioconductor has some functionality for working with them. So what is a FASTA file? They've been around a while. FASTA format. So FASTA format, it's a text-based format for representing sequence data. That could be A, Cs, Ts, and Gs data if we were talking about DNA, or it could be amino acids if we're talking about proteins. And all it is a text file, but it has a predictable format. Might look like this. So if you open a FASTA file, often you'll have lots of entries. You'll have one entry per sequence that you want to store. And each entry has to follow a particular format. So this would be one entry in a possible FASTA file. It starts with descriptive information. Here's what I'm providing you a sequence for. So this tells me about what the gene is, what the organism is, and what we know about this gene, if anything. And then what follows is the actual sequence. So I uploaded a FASTA file to eCampus, and I'll show you how to read one in. This comes with seq-in-R, this FASTA functionality to read or write a FASTA file. let me download an example. So under Data, Sequencing, and I want the FASTA file for the Mike gene. Mike gene FASTA. Mike gene, there's a Mike gene and there's a Mike protein. I'll go ahead and download both. The Mike gene file has DNA sequence, and the Mike protein has amino acid sequence. All right, and let's open one of these up. I'm just going to open it in my text browser because it is just a text file. And here's what we have. So we have a descriptive line. We don't have to know what all of this means, but it's telling me, key basic information about this gene. Where did it come from? Where is it located? Chromosome 8 in this case. Which species are we talking about? And so on. I think this is the actual region on the chromosome. Like start at base, whatever that is, 1.2 billion or something. So we have a single sequence in this FASTA file, which I think they have. I don't think you want spaces, empty lines. If there were multiple entries, it would look like this, and then down here, I would have a space and my next entry. Like that. So typical with a modern data set to have thousands and thousands of entries in a FASTA file. It's just a convenient, standardized way to represent and pass along to store sequencing data or sequence data. Even between lines like this? Oh, okay. So I guess the key is that it has to recognize the caret thing. There is a function for reading in a FASTA file, and then you can just get the sequence out of it. So I'll just run this real quick. I'll skip the protein one. It's the same exact thing, except it gives you amino acids. All right, so before I run this, it's going to go to a specific location on the computer to find the data. So I need to make sure I've got things set up right. Let me rename this. That's right. No. Seek analysis data. All right, so I want R, the workspace, the working directory, to be one folder up. Change directory. Nathan, my documents. Let's do. Okay, so now I'm in script, so if I go back one, I think that will work. So let me try this. What is the class? It's a list. I'm going to use the getSequence function to print it out. So there's the actual ACTG sequence. So if somebody passes you a FASTA file, even if it has thousands of entries in it for all these different sequences, like in a next generation sequencing application, you could use seqnr and the read.fasta function to pull that in. What might you want to do with sequence data? We'll talk about this in a minute, but what if we just tabulated all the different letters? I did table. If you do table for an object in R, it just tabulates how many times did you have this particular value. And in this case, we had 1,191 A's, 1,426 C's, and so on. Very basic way to characterize a sequence or a whole genome would be, what are the proportions of the letters? And I'll show you in a second that, down here, we'll get to this next time, the proportion of G's and C's, interestingly, is biologically relevant. So out of the 5,366 letters, what proportion of them are either a C or a G? Obviously easy to calculate right now because we have the whole sequence. But that turns out to be biologically interesting because it can have chemical ramifications. If there's lots of C's and G's, it can change the chemical characteristics of the gene. Comparing two species in terms of C and G content can be informative because it could tell you about functional differences between those species. I will end here. I'll go ahead and read in the protein file and show you one more thing. So this is same exact code, except that I'm asking for mic_protein.fasta. And I specified there at the end of the call. Up here I said seq type equal DNA. So I'm saying what's in my FASTA file is A, C's, T's, and G's. Now I'm specifying amino acids. So it's expecting something different. And we'll get the sequence. Looks like that. So we had 5,000 something A, C's, T's, and G's. We have 450 amino acid letters. Just to make sure you guys were listening the other day, would you expect to have more amino acids or more A, C's, T's, and G's? Okay, why? Okay, good. Two things I would say. Here's all of the A, C's, T's, and G's. Some of that's probably going to get cut out because a lot of that is probably introns. We're not going to code that into protein. Suppose that after you cut out all the introns, you're left with, well, you tell me. After we cut out all the introns, approximately how many A, C's, and T's, and G's do you think we had left? Okay. Mentioned this just in passing, so it's fine if you don't remember, but A, C's, T's, and G's, once it's been chopped up into all exons, once it's been chopped up into that, this process of translation, remember, starts at one end and starts reading, and it reads in triplets. So we looked at a table that said if you have an A, C, G, that corresponds to amino acid such and such. So here I have letters that are amino acids. Each one of these is a triplet of A, C's, T's, and G's. One thing, let's look at the sequence composition of the amino acids. I'm going to table this up. And now it's a little more interesting because I don't have just A, C's, T's, and G's. These are all the different amino acids that are floating around. How many of them are there? There's 20. Depending on which resource you look up, there are 20 basically 20 protein-forming amino acids in humans. Correct me? Because I've seen different things. There's some amino acids that go into proteins. There's some amino acids I think that don't. But I think it's fair to say there's 20 of these letters for humans. Yes, these are initials for amino acids. Yes. Some of the and I think there can be special codes, like if they don't know, if they know it is either A or C or something, they might use one letter to indicate that. But for the most part, if you Google genetic code, you see what the correspondences are. So if there is a AGA, that's arginine, which is given by the letter A in the, this is the second letter. So actually I'd have to look up exactly what the single letter correspondence is. I'll find it. But that's what these correspond to, these letters right here map back to, what's that? It's for tryptophan, okay. These codes, yeah. Okay. So if we need it, we'll figure that out. I don't think there's going to be any need for you guys to translate these into actual amino acids in this class. It's easy to find, though, if you want to map them back. Okay. The last little bit here is just showing how to use the corresponding function write.fasta. So if you have your own sequence data, you can create a FASTA file for sharing. Okay. And next time we will start talking about sequence alignment. It's an interesting problem. What's the optimal way to line up these two sequences if you reward matches with this and you penalize mismatches with that and so on? Throw the computer at it and try to find the best alignment. Okay, questions? What kind of files? I'll have to look them up. In what context do you? And what is in the .quad? Quan or quad? Okay. Okay. We'll see some example next-gen sequencing data, and I'll see if I can work that in there somewhere. All right. See you Wednesday.